Welcome back. We are now on Part B, which is of Chapter 21, and this includes the adaptive body defenses, or the adaptive immune system. This part of the immune system is a specific defensive system which eliminates almost any pathogen or abnormal cell in our bodies. It does this by amplifying inflammatory responses and by activating complement. The adaptive immune system has one shortcoming. That is, it must be primed by an initial exposure to a specific pathogen first before it can work, and this priming takes time. Adaptive immunity is specific, meaning it recognizes and targets specific antigens. It's systemic, so it is not restricted to the initial site. And it has memory, meaning it mounts an even stronger attack to known antigens during the second and subsequent exposures. There are two main branches of the adaptive immune system, and they are the humoral, or antibody-mediated immunity, and the cellular, or cell-mediated immunity. Beginning with humoral immunity. It involves antibodies produced by lymphocytes, which freely circulate in body fluids. Antibodies bind temporarily to the target cells, making it temporarily inactive. The target cell is now marked for destruction by phagocytes or complement. Humoral immunity has extracellular targets. Cellular immunity involves lymphocytes acting on pathogens. They directly kill infected cells and indirectly release chemicals to enhance the inflammatory response, or they can activate other lymphocytes or macrophages. Cellular immunity has cellular targets. Antigens are substances that can mobilize adaptive defenses and provide an immune response. These are targets of all adaptive immune responses. Most are large, non-self molecules. They can be a complete antigen or an incomplete antigen, called a hapten. They can contain antigenic determinants and can also be a self-antigen. Let's go through what we mean by these characteristics. When we say an antigen can be complete or incomplete, a complete antigen has two important functional properties. It has immunogenicity, which is the ability to stimulate proliferation of specific lymphocytes. It also has reactivity, which is the ability to react with activated lymphocytes and antibodies released by immunogenic reactions. Examples of complete antigens are foreign proteins, polysaccharides, lipids, and nucleic acids, which are seen on such things as pollen and microorganisms. Incomplete antigens, also called haptens, involve molecules too small to be seen, so they are not immunogenic by themselves. For example, small peptides, nucleotides, some hormones. Haptens may become immunogenic if the hapten attaches to the body's own proteins. The combination of the protein and hapten is then seen as foreign. A hapten attached to a self-protein causes the immune system to mount an attack that is harmful to the person because it attacks self-proteins as well as the hapten. Poison ivy, animal dander, detergents, and cosmetics are all sources of haptens that attach to a self-protein, leading to the body to attack the self-protein. Antigenic determinants are parts of the antigen that antibodies or lymphocyte receptors bind to. Most naturally occurring antigens have several antigenic determinants that mobilize several different lymphocyte populations and form different kinds of antibodies against them. Large, chemically simple molecules like plastics have little or no immunogenicity. Figure 21.7 illustrates how most antigens have several different antigenic determinants. We see this red antigen has 3 antigenic determinants. You see them here. This results in 3 types of antigen binding sites and 3 different antibodies that will attach to it. Self-antigens are cells that are covered with proteins which are not antigenic to oneself, but may be antigenic to others in transfusions or grafts. Major histocompatibility complex proteins, MHCs, are important self-proteins. They contain a groove that can hold a piece of a self-antigen or a foreign antigen. T lymphocytes can recognize only antigens that are presented on MHC proteins. Let's say that again. T lymphocytes can recognize only antigens that are presented on MHC proteins. Adaptive immunity involves 3 types of cells. B lymphocytes or B cells, which are involved in humoral immunity. Excuse me. T lymphocytes or T cells, which are involved in cellular immunity. And antigen-presenting cells, or APCs, which play essential auxiliary roles in immunity. Beginning with lymphocytes. Figure 21.8 explains lymphocyte development, maturation, and activation. T and B lymphocytes share a common development. Both lymphocytes originate in red bone marrow. Lymphocytes become educated and mature in primary lymphoid organs, the bone marrow for B cells and the thymus for T cells. Immunocompetence is when the lymphocytes can recognize only one specific antigen. B or T cells display only one unique type of antigen receptor on their surface when they were mature, allowing them to bind to only one type of antigen. Self-tolerance means that lymphocytes are unresponsive to the body's own antigens. The T lymphocytes maturation occurs under positive and negative selection, as seen in Figure 21.9. The positive selection process selects T cells capable of recognizing self-MHC proteins. The T cells that can't recognize self-MHC proteins are destroyed by apoptosis. Those T cells that do recognize self-MHC proteins proceed to the negative selection process. So first we have the positive, and if they survive this, then they get to go to the negative. Negative selection makes sure that T cells do not recognize self-antigens result in apoptosis, eliminating self-reactive T cells that could cause autoimmune disease. This process is called clonodeletion and ensures self-tolerance. T cells that do not bind to self-antigens result in survival and continued maturation. All right, so we have to get through the positive selection. So we want to be able to recognize the self-MHC in the positive selection. If it recognizes that, then it goes on to the negative selection. In the negative selection, the T cells must not recognize self-antigens. Okay, so you want to recognize the self-MHC, but not the self-antigen. All right. Excuse me. Not much is known about B cells maturation in the bone marrow. Only immunocompetent B cells are allowed to mature. Those are self-reactive and eliminated by clonal deletion. Immunocompetent but still naive B and T cells leave the thymus and bone marrow. They seed the secondary lymphoid organs and circulate through the blood and lymph where they have a better chance of encountering an antigen. When a lymphocyte's antigen receptor binds to an antigen, the lymphocyte develops further. Clonal selection is when the lymphocyte is selected to differentiate into an active cell by binding to its specific antigen. If the correct signals are present, the lymphocyte will complete its differentiation into an active cell. Once selected and activated, the lymphocyte proliferates, making exact copies of itself referred to as clones. Most clones become effector cells to fight infection, but a few remain as memory cells. Memory cells enable the body to respond more quickly to the same antigen the second time it's encountered. B and T memory cells and effector T cells circulate continuously. Genes, not antigens, determine which foreign substances the immune system recognizes. The variety of immune cell receptors are a result of acquired genetic knowledge of microbes. There are approximately 25,000 different gene codes for up to 1 billion different types of lymphocyte antigen receptors. To illustrate that exposure matters, we now know that small children exposed to dirt and the outdoors have better outcomes than those kept in an antibacterial environment by protective parents. Table 21.4 compares B and T lymphocytes. Make sure you spend some time and review this.